Okay, start with chapter 3 now. Chapter 3 is mood disorders. We're going to be covering major depression and bipolar disorders. So, first of all, when you're talking about any disorder, not just mood disorders, you have to think, okay, how do we define this? How is it that we're going to define what constitutes a depressive disorder? And that's a huge problem with pretty much every disorder almost. I mean, some of them are real obvious, like schizophrenia. But with depression, it's very different because the symptoms are very subjective. It depends a lot on the resiliency of the person who's suffering, their environment, what's expected of them. There's just it's very subjective. It's very hard to come up with a very objective criteria for determining what constitutes depression. Because most of the time, not most of the time, there just really isn't like something like a blood test that you could take for depression. You diagnose it through patient interviews and through patient history and records of patients' behavior. I mean, that's about it. So the diagnosis ends up being a very subjective thing. And the reason that's one of the many reasons that's a problem in pharmacology is because you may be giving a drug to treat a specific condition, person after person after person. So you give drug A to treat psychiatric condition D in patient 1, then in patient 2, then in patient 3, etc. And you're assuming that drug is going to do the same thing in all of these patients because you have diagnosed them with the same thing. But in fact, maybe you didn't diagnose, maybe they don't have the same thing, right? Maybe each individual's brain is different somehow, and that would lead to the very wide variety of responses that we see to antidepressants. So the good old DSM comes in handy, the Diagnostic and Statistical Manual of Mental Disorders, and they of course have a set of criteria for depression, and they're listed here on this slide. Depressed mood for most of the day. If you think about it, what does that mean, right? A depressed mood for one person may not be the same experience for another person, right? A diminished interest and pleasure in most activities, right? So that's pretty easy to measure. A significant change in appetite and weight, also pretty easy to measure. Insomnia or hypersomnia, right? So I love this one. You're either sleeping too much or too little. It affects your sleep. It can affect your sleep, I should say. Motor agitation or retardation observable by others, right? So either you're just kind of always bouncing around and fidgeting, or you're just completely just dead, right? Not moving. Diminished energy and fatigue. A diminished ability to think or concentrate. This is a big one for me. This is how, in my own experience with major depression, this and the next one are the two that pop up immediately when I'm first starting to feel, get depressed. Diminished ability to think or concentrate, and feelings of helplessness, worthlessness, or guilt. And then finally, recurrent thoughts of suicide or attempted suicide. And of course, this last one is the reason why depression is so dangerous, because depression can literally kill you. Depression, people with depression will get locked into a frame of thinking that leads down the path of suicidal ideation and then perhaps ultimately suicide. Now, most of the evidence that we have today says that we are dealing with a broken serotonin or noradrenaline receptor system. And that brokenness is what's causing major depression. Okay, so your major depression is due to a dysfunction in your serotonergic, your serotonin, or your noradrenaline systems, and it's affecting all of the other systems that those major systems project to. One of the many bits of evidence for this monoamine hypothesis, and it's called the monoamine hypothesis because serotonin and noradrenaline are monoamines, meaning that they are molecules that only have one amine group, monoamine. So the monoamine hypothesis, one of the small bits of evidence for the monoamine hypothesis is that I can give you a drug that depletes the availability of neurotransmitters, and it will cause you to experience a lot of symptoms of depression. So I can give you a drug like reserpine. Reserpine is a drug that will go into your serotonergic and noradrenergic neurons and basically make them leaky. It basically causes the vesicles to release their neurotransmitter and just spill everything. So that there's no neurotransmitter to use to signal the other neuron in the across the synaptic gap. So this and a bunch of other evidence has, over the years, since about the mid-'80s, has been used to establish the monoamine hypothesis of major depressive disorder. So here is a molecule of serotonin, and you will see the single amine group. Amine, the word amine always implies nitrogen. So there's a nitrogen bound to two hydrogens at the end of that carbon chain. And you'll see on the other side there's a ring of carbon atoms right here, and with hydroxyl groups all bound to them. So that's what we mean by monoamine. Now, there have been a lot of problems with the monoamine hypothesis. As a matter of fact, the thinking today is that it's not quite right. And these next two slides in this lecture are probably going to be some of the most important bits of information you're going to get in this course, because they are going to cover why the monoamine hypothesis may not be correct. And there's a lot of evidence that suggests it may not be exactly right. For one, whenever I give you an antidepressant, so let's say for the sake of argument, you're suffering from major depressive disorder. I've diagnosed you with major depressive disorder, and I prescribe you Prozac. And Prozac is a serotonin selective reuptake inhibitor, as you recall. So it's going to be inhibiting the uptake of serotonin, and therefore increasing the availability of serotonin in the synaptic gap. And of course, the serotonin, the theory goes, the serotonin is enhanced. The activity of serotonin is enhanced, and Voila, you feel better. Well, the problem is that if you start taking Prozac pills, and let's say it's a relatively high dose. Let's say I'm giving you, I don't know, 80 milligrams a day, right? If you start taking Prozac every day, within a few hours of taking your first dose, I would be able to measure increases in plasma concentrations of monoamines, like serotonin. So it works almost immediately, right? Within a few hours of taking the drug, your brain is producing or maintaining more serotonin in the synaptic gaps of the brain where serotonin is used. However, famously, that doesn't translate into feeling better. For some reason, it takes anywhere from 2 to 6 weeks for people to start feeling better, to start feeling less depressed after taking a daily dose of Prozac. So why is that? Why is it that taking the Prozac, it's doing its job right away in the brain, but you don't feel better for another few weeks, right? Why is that? And that's been a big question mark for a very long time. So now the newest hypotheses has to do with neural degeneration. So there's a degeneration of neural innervation in the hippocampus and the frontal cortex, caused by the monoamine neurons not producing enough growth factors to keep the connections going in the hippocampus and the frontal cortex. Okay, so apparently these monoamine neurons aren't failing because they aren't producing enough monoamine. Rather, they're failing because they're not producing enough growth factors to maintain the growth of neurons in the hippocampus and in the frontal cortex. So it's still kind of related to monoamine neurons, but it's not just the neural transmission of monoamines. It's not just the neurotransmission that's controlled by monoamines. Rather, it's something else, the use of growth factors by these neurons. So what happens is the monoamine activity gets reduced because the neurons are being inhibited by their autoreceptors. So there's receptors on the neuron that sends the neurotransmitter. There are receptors on that neuron that detect the amount of neurotransmitter in the synaptic gap. And those guys get inhibited, which means the cell can't tell how much neurotransmitter is in the synaptic gap, so it goes down. One of the other reasons why we've gotten better at revising this hypothesis is because we have better and better tests for, I should say, we have better and better models for depression. Chronic mild stress, like putting a rat in a shock chamber and just giving it like low-level foot shocks randomly, forced swimming, and removal of their olfactory bulbs have been used to effectively induce behavioral depressive symptoms in animals. And those types of models have become slightly better for us to evaluate the effectiveness of drugs, and also to see and understand the neural degeneration that might be associated with the monoamine neurons. So the amine hypothesis of depression says that if you deplete the neurotransmitter and serotonin activity, you're going to get depressed. The first evidence, as I mentioned earlier, is depletion by reserpine. Reserpine is the drug that causes the neurons that are using serotonin and norepinephrine to release their serotonin and norepinephrine and get rid of it, so that it basically makes the neurons leaky, so that they just spill out all of their serotonin and it's not used. Animal studies where you deplete norepinephrine and serotonin in various ways, either pharmacologically or surgically or genetically, also support this idea of depression be underlying a monoamine dysfunction underlying depression. Effectiveness is medication. So, there are a lot of people, myself included, who have gotten better with these drugs that increase monoamine function, right? And then autoreceptor knockout mice. So I can actually create a mouse that does not express the autoreceptor gene, and they are very susceptible to depression, right? So the underlying mechanism that is behind these effects, these amine effects, may be related to a compound referred to as BDNF, a brain-derived neurotropic factor. And whenever they use the word factor, they mean like some chemical, some compound, some chemical, right? That does something very complex. In this case, the brain-derived neurotropic factor is a nerve growth factor that is used by the brain for normal cell survival and normal cell growth. Right? So it keeps the cells alive, helps you express receptors, and helps new neurons spring forth. Okay, so it's pretty important. If you don't have BDNF, you've got a problem. The synthesis of this neurotropic factor is dependent on a series of proteins working together. The first thing that happens in this chain of events to create a neurotropic factor is the influx of calcium ions when a monoamine metabotropic receptor is activated. So whenever one of these, remember what a metabotropic receptor was? It was those receptors that are connected to other proteins. So instead of just changing shape and letting ions in or out, what a metabotropic receptor does is it is coupled to other proteins to start the dominoes falling on some type of postsynaptic activity. In this case, whenever the monoamine metabotropic receptors are stimulated, calcium influx, it causes the influx of calcium into the neuron with these receptors. And that is the first domino that falls to begin the creation, the chain of events that ultimately ends in the creation of brain-derived neurotropic factor. So in other words, if you block your monoamine receptors or don't have enough of them, you're not going to be influxing enough calcium. Therefore, you're probably not going to be producing enough brain-derived neurotropic factor. Okay, so that's the connection. And here's a awful little picture of the process. So right here, this little funnel thingy right here, this upside down funnel, is the presynaptic neuron, and it's these little tiny little red dots right here are the monoamine, like serotonin, for example, or norepinephrine. So the little red dots bind to these little tiny little blue guys right here, which are the receptors in this big cell, right? Now, what happens is the receptors, it binds to the receptors. Whenever the receptors change shape, it causes an influx of calcium ions. The calcium ions are used in the activation of cyclic AMP, which is a second messenger for PKA. And then PKA then goes and increases, is used to create CREB, C-R-E-B, inside the cell nucleus, which ultimately leads to the upregulation or creation of the brain-derived neurotropic factor. So the binding of the neurotransmitter like norepinephrine or serotonin begins this chain of events that ultimately leads to the increase in the production of brain-derived neurotropic factor. So that's kind of cool. That's a very, very, very interesting pathway. So to continue, the role of the brain-derived neurotropic factor, the calcium influx, the influx of the calcium ions activates the second messengers, which are those proteins that are, comment in the arrows afterwards, right? Activate second messengers which regulate the transcription of BDNF from the cell's DNA. So all of these guys here, what these guys here do, the PKA and the CREB, these are second messengers that basically tell the cell to produce more brain-derived neurotropic factor by activating the transcription of their DNA that codes for this particular protein. The second messenger that's mediating the brain-derived neurotropic factor is a protein called CREB. So CREB is what actually activates the transcription of BDNF. And CREB stands for cyclic adenosine monophosphate. Cyclic adenosine monophosphate, response element binding protein. So CAMP, response element binding protein. And that's basically what that means is when this thing is activated, when the CAMP response element binding protein is activated, it binds to another protein that is responsible for mediating the transcription of the genes responsible for generating more BDNF. So the bottom line is that if you downregulate your monoamine neurons, so in other words, if you reduce the number of monoamine neurons, that's what downregulation means, is pulling some of those, monoamine neurons, right? The downregulation of monoamine neurons, so getting rid of some of your monoamine neurons, leads ultimately to further downregulation of the growth factor in neurons in the hippocampus and the frontal cortex. Now, if you recall in I think it was the first lecture where we were going through brain anatomy and I was showing you the projections of different neurotransmitters. So if you recall, norepinephrine and dopamine, they kind of start in the lower parts of the brain and project up into the frontal cortex and into the hippocampus. Okay, so that's what they do. That's their job, is to project into those areas and mediate our affect, our mood, our behaviors. But if they're downregulated, if the monoamine neurons are downregulated, then the cells that live in the hippocampus and the frontal cortex, well, they quit producing this essential growth factor. Why do they quit producing it? Because that little chain of events there that begins with the calcium ion influx isn't happening anymore. Because that calcium influx is kicked off by binding of a monoamine to its receptor. So if that ain't happening, there's no BDNF being produced in the hippocampus or the frontal cortex. So this downregulation disrupts the signaling. So basically the neurons aren't being signaled as much, and you're not growing any new neurons. So it's really hard for you to adapt and deal with stressors, right? And ultimately it leads to degeneration of the neurons. So if you think about it, this makes a lot of sense, because it would help to explain why it takes 2 to 6 weeks for you to get better after you take a neurotransmitter, right? Because what's happening is, after you take an antidepressant, because the antidepressant is sort of, in an offhanded way, trying its best to stimulate what few receptors you have left for the monoamines, but it takes a while for it to build up the number of receptors, and it takes a while for the BDNF chain of events to start up again. So that's why it takes 2 to 6 weeks for you to get better from taking antidepressants. This is a weird slide. It's just kind of messy. So how do depressed patients get downregulated BDNF transcription and the cascade of neuronal consequences resulting from it, right? Well, basically this question is asking, okay, if I'm a depressed patient, why is my BDNF getting downregulated, right? Why is it going away, right? Here's the problem. You can have two people exposed to the exact same stressors, and yet they're both going to have one is going to get really depressed over it and downregulate their BDNF and get majorly depressed. The other one's going to be fine. They might have some problems, but for the most part, they'll be okay. They don't get as depressed. So what's going on? Why does this happen? Well, there may be, for example, genetic predispositions in the big machinery of proteins working together to regulate your growth factors and your monoamines. There may be some slight subtle differences between individuals in how much protein they're expressing to do all of this intricate work, or maybe there's some proteins missing, etc. Basically, we've done these studies with animals that express the proteins, the different proteins associated with the production of BDNF. So I can create a rat or a mouse that doesn't have the Krebs protein, for example. Or I can create a rat or a mouse where I can turn on or off the Krebs protein at will. And whenever that happens, the BDNF gets downregulated, it goes away, and that animal starts feeling depressed. And then if I give it antidepressants, it starts to feel better again. So the relationship between brain-derived neurotropic factor and stress is this. Now, remember this. It has always been understood that there is a relationship between stress and depression. As a matter of fact, at a very high level, most people consider depression to be an adaptive reaction to stress, right? So what happens? Well, at the hippocampal and frontal lobe level, you've got those two brain structures processing information. And one of the things that these two brain structures can process is information about stress. So here comes the stress, which is this star here. The stress then directly affects the BDNF pathways. by reducing neuronal survival and plasticity, and ultimately leading to a depressed mood. By contrast, I can give somebody an antidepressant treatment, right? And what this is going to do is increase cell growth and survival, leading to an increase in elevated mood, right? So this is a very simple diagram illustrating the relationship between the brain-derived neurotropic factor, which is going to mess up your brain by reducing neuronal survival and plasticity, meaning the growth of new neurons, and ultimately leading to a depressed mood and activity. Whereas antidepressant treatment is going to help increase cell growth and survival in a roundabout way, and ultimately leading to an elevated mood. And you can see here, one of the things that you can see, and this is, when I first learned about this really freaked me out. But what you see here is a quote-unquote normal brain versus a brain with depression. And this is a coronal section. So basically it's where, I'm taking, the knife is going like this. Right, the knife is cutting the brain like this, and then I remove this part of the brain, then you're looking at that flat surface, okay? And that cut is happening right about here. Oh, well, I'm trying to see where the camera is. Okay, yeah, right about here, I guess. And so here's the top of the brain right here, bottom of the brain, left side. Oh, shoot, sorry. Left side, or right side, left side. And of course the hippocampus curls in 3 dimensions, like this, right? It kind of curls. And this is the hippocampus right here on each side. Kind of the main body of the hippocampus right here, right next to the temporal lobes. And this is a close-up, right? These dark areas are the hippocampal areas. The hippocampus is a very, very complex organ. It's really complex. And so it's real easy to see when you're looking at a brain slice. The hippocampus is like one of the easiest things to find on a brain slice. So here it is here and here in a normal person. But remember, a depressed person has reduced BDNF, which means they're going to have reduced neuronal growth. So you're going to see an atrophy, a reduction in volume in areas like the hippocampus. And sure enough, that's what you see here. This is the brain of a depressed patient. And one of the things you notice is this, you don't see this nice little sort of like oval shaped area here. Instead, you just see like this line here, which is all that's left of the hippocampus. So in a very severely depressed patient, that's what you end up with. So the revision, right? The revision of the monoamine hypothesis, we have a direct measure here of BDNF levels, brain-derived neurogrowth factor levels. On the y-axis, you see the BDNF as measured in picograms per milliliter of plasma. Right, so it's pretty low. It's pretty hard to measure this stuff, but there it is, going from 0 to 900 pg per ml. Right, and then across the x-axis, you see the different groups in this bar chart, along with the error bars. This is normal people who are not depressed, and they got a ton of BDNF. These people here are MDD stands for major depressive disorder. MDD, major depressive disorder, and these are people that have reported it for the first time in their lives. And you'll notice they have significantly decreased levels of MDD relative to the normals. This is the plasma concentration of BDNF in major depressive disorder patients where where it's recurrent, where the depression just keeps coming and keeps coming and keeps coming and keeps coming. Okay, and then finally, this is the major depressive disorder, multiple diagnoses, it's a major depressive disorder plus individuals with severe suicidal ideation. And you'll notice that these two bad ones, the lowest ones here are these two here, the folks with recurrent major depressive disorder and the folks with major depressive disorder plus suicidal ideation. So it's a nice demonstration, a very simple demonstration of how important the brain-derived neurotropic factor might be to underlying depression. We have another bit of evidence here, right? So on the, and this is basically the effects of antidepressants on immobility in a forced swim test with rats. So basically, normally, if you just take a regular rat, a normal regular rat, and put them in this forced swim test where they're like in a little tub about yay big, right? About yay big, and it's very deep, about that, about maybe 2 feet of water. And you put them in this tub, and there's no way for them to get out, and just let them swim and swim and swim and swim and swim until they're tired. Once you take them out and dry them off, and then put them in their cage, they tend to just sit there and just like not move. And it's not because their body aches or they're tired per se. It is because of a depressed mood, right? Because we've tested them by giving them we've put them back in their cage and give them some analgesics to help with body pain, and you can give them stimulants to stimulate their movement, and they don't respond. So they're a bit depressed. However, one thing you can give them is an antidepressant. And what you see here is the post-forced swim rats that have been given an antidepressant versus not treated with anything versus an anti-anxiety drug. That's what an anxiolytic drug is. It's an anti-anxiety drug. And you'll see that the no treatment and the anti-anxiety drug rats spent a bit more time being immobile relative to the antidepressant rats. It is a significant difference. It's a relatively small difference, but it is significant, which tells us that the antidepressants are having some sort of effect in this depressive cascade of proteins being activated as a result of stress. So, how do we treat major depressive disorder? Well, for a long time, there wasn't a treatment. It was kind of talk therapy and I guess alcohol self-medication. It's been the traditional treatment in my family for generations. But the first drugs that were released that seemed to be effective against major depressive disorders came about in the late 1950s, like 1958, 1959, during the Cold War. Now, the thing about these drugs is they were sort of discovered accidentally, A, and B, they had some severe side effects. But nonetheless, they seemed to be effective in treating depressive disorder and started being prescribed for that. Even though they didn't know how they worked. It's kind of like aspirin. Aspirin's been around for 150 years, something like that. And for the first 100 years or so of aspirin being used, we didn't know how it worked. We had no idea until we started discovering prostaglandins and COX-1 and COX-2 receptors and things like that. We were able to understand how aspirin is, the mechanism of action of aspirin. And similarly, there was no understanding of how these guys were working except to say, well, they work, right? However, in the 1960s, a lot of work was being done, mostly in animals, to identify all of the monoamine neurotransmitters and the pathways, the projections of the neurons that used monoamine transmitters. And so in the 1960s, we started to understand where these pathways were going, what these neurotransmitters were doing, and started to make the link between the neurotransmitters and the drugs that are being used to help treat depression. These drugs that were created in the late 50s and 60s were known as the tricyclic antidepressants. They're called tricyclic because when you look at the molecules of each one of these different types of drugs, it has three carbon rings, so tricyclic, and that's how they were named. And their ability to treat major depression was discovered very much by accident, because these drugs were initially used as gosh, I want to say they were being used for I cannot remember. I'm blanking on what they were originally being used for. I want to say seizures. I know. I can't remember. Anyway, they were being used for some purpose, right? Experimentally, by the way, experimentally. and and then some clinicians started noticing that when folks were taking these for a while, they started to get less depressed. And it's like, oh, okay, interesting. So that's kind of how that's how that came about, right? Quite by accident. And here's a picture of two of the tricyclic antidepressants that are out there. This top one is imipramine. So you'll see the three rings, 1, 2, and 3. And the middle ring has a nitrogen atom, and that nitrogen atom is connected to a carbon chain here. And a very similar drug is amitriptyline, which is this guy right here, which again, instead of having the nitrogen atom, it has a double bonded carbon, which has a nice little chain of carbohydrate chain here. Oh, no, not carbohydrate. There's no O's, but carbon chain here. That's why they're called tricyclics. And here is a list of the tricyclic antidepressants that have been around for a long, long time. So here you have the generic name, drug name, and then here you have the trade name that was given by the company that manufactured them. So the big ones back in the day were Elavil, which was amitriptyline, right? Tofranil, which was imipramine. Desipramine, which was norpramine, right? So those were the big ones, and then we had a bunch of others here. Sermontil, which was trimipramine, Triptyl and Vivactyl, which was protriptyline. All of these drugs are different types of tricyclic antidepressants, and they were prescribed for many, many years. Here in the final column, you see the half-life of some of these drugs in hours. And you'll notice that there's a lot of variation here. So, for example, protriptyline, you'll see its half-life is 74 hours. That's a long time, right? That's a really long time. Whereas, with imipramine and Elavil, their half-life is from 10 to 28 and 10 to 20 hours. So there's quite a variety. There's quite a difference in half-life there, which is going to lead to differences in prescribing it. But these are your first batch of antidepressants that came out in the 1950s or really in the 1960s. So how do they work, right? What did these guys do? Well, the tricyclics were reuptake transporter protein. They bound to the reuptake transmitter. They bound to the reuptake transporter proteins for both norepinephrine and serotonin. Okay, so basically they would go into your brain and they would sit on top of the reuptake proteins in the synaptic gap for norepinephrine and serotonin. And what that would do is it would keep the norepinephrine and serotonin in the synaptic gap longer. So those two neurotransmitters could not be reuptake, sucked back into the neuron where they came from. So they would stay out in the synaptic gap longer, and they could just kind of bind and rebind to the postsynaptic receptors and to the presynaptic receptor. They could just bounce around in there, binding and binding and binding and binding. So what does imipramine do to the BDNF in the hippocampus, right? Well, imipramine has a nice effect of significantly increasing, slightly increasing. It's not a huge effect. But what we have here is on the y-axis, you have the percentage of control. In other words, percentage of the of whatever you're measuring. In this case, we're measuring BDNF messenger RNA relative to a control group, right? Relative to a control group. And here we have the control group in the light gray and the imipramine group in the dark gray. These four sets of bars here, 1, 2, 3, 4, they represent an area of the hippocampus. So the areas of the hippocampus are designated as, they have different names associated with them. So there's the CA1, the CA2, CA3, the CA4, and then the dentate gyrus, which is a part of the cortex that sits right next to the hippocampus and is usually considered part of the hippocampus. So in CA1, CA3, CA4, and in the dentate gyrus, you see there is a slight effect, significant but slight effect of the imipramine on the presence of messenger RNA for BDNF. Now, as you recall, messenger RNA is the type of RNA that is used to communicate from inside the nucleus to the ribosomes inside of the cell body. Messenger RNA carries the RNA signature, the DNA signature of a protein, in this case BDNF. It carries it out so that it can be built, so the protein can be built in the cell body. And so more messenger RNA of a particular protein means that presumably you have more of that protein. So what this is telling us is that imipramine increases the amount of protein, of that protein, BDNF, relative to a control group in the hippocampus, which is very cool. And here is these are really neat little fluorescence studies. Basically what they do is look at the presence of particular proteins, and by having them express a section that can be lit up in using a particular microscopic technique. And then when you expose these specialized genetically prepared animals to this particular technique, you can see the expression of the protein in each brain slice. And here you see the expression of BDNF and the growth factor here in the hippocampus. Now, one of the big problems with tricyclic antidepressants is that they had pretty significant side effects. That was one of the really attractive things about Prozac when it first came out in the late 80s and early 90s. When it first came out, and that's one of the reasons it became such a big moneymaker for Eli Lilly, was because it was an antidepressant that didn't have as many severe side effects as the tricyclic antidepressants do. So some of these side effects included, first of all, the tricyclics are great antihistamines, they're antihistamines. They block the histamine receptors in your brain, your whole body, I guess, but in your brain is what we're concerned with, right? And what happens when you take an antihistamine? You get drowsy and you get fatigued, right? And sure enough, that was a huge side effect of the tricyclic antidepressants was this drowsiness. By the way, I shouldn't say was a side effect. These drugs are still around. Tricyclic antidepressants are still around today. There are some people whose depression does not respond to anything else but a drug like imipramine or amitriptyline, and they're taking it for their depression. So they're still around today. If you're like an insomniac with your depression, if your sleep disorder makes you agitated and insomniac, then you probably welcome this histamine blockade, because if you have insomnia, then if you take something that makes you sleepy, that's a good thing, right? So there were a lot of patients actually kind of liked it, right? You can actually overdose with tricyclic antidepressants. That was another problem. It's really hard to kill yourself with Prozac. It's really hard to overdose on Prozac. I mean, if you take too much Prozac, you're going to be really sick. But you're probably not going to die. However, with the tricyclic antidepressants, you could die from severe seizures, cardiac arrhythmia, hypotension, where your blood pressure just completely drops, and ultimately possibly death. So it is possible to seriously hurt yourself with an overdose of tricyclic antidepressants. Another type of related neurotransmitter is the MAO inhibitors, monoamine oxidase inhibitors. Monoamine oxidase is a compound that is a compound located in the presynaptic, in the synapse, right? The area between the presynaptic and the postsynaptic, where the neurotransmitters travel through, right? MAO is a protein that's located there, and what it does is it breaks down the monoamines. It oxidizes them, breaks them down so they can be recycled. Now, if you inhibit this protein, if you inhibit monoamine oxidase by taking a monoamine oxidase inhibitor, what does that mean? Well, it's not going to be doing its job anymore. It won't be breaking down serotonin and norepinephrine, so the serotonin and norepinephrine will stay around longer, right? And it was again discovered accidentally, as so many of these things are, that for some people, these things work okay as an antidepressant. However, they have some severe side effects. If you block monoamine oxidase from being produced, you're also going to be blocking a lot of other important proteins that help you metabolize different phenol-related compounds. So when you take these drugs, you have to be really careful about what you eat, because you might be overdosing on some particular compound that you cannot metabolize, and you might end up building up a dangerous amount of this compound in your body and end up hurting yourself very badly, right? So they are very dangerous drugs. They're not used that often anymore. There are a few individuals out there who maybe will take a low dose in combination with their other antidepressants that might help them their particular depression. But for the most part, these drugs aren't used too much anymore, but they still exist. So the way the monoamine oxidase is worked is so all of the monoamine neurons, like the serotonin neurons and the norepinephrine neurons, produce MAO, monoamine oxidase, to deaminate. In other words, to remove the nitrogen group from the neurotransmitters in the terminal button. And basically, it helps to regulate the amount of monoamine available for storage and release. And as with the other drugs that affect norepinephrine and serotonin and monoamines, it takes a while for the MAO inhibitors to show an effect. Right, it takes 2 to 6 weeks. And like I said, they have some pretty significant side effects. Sedation, fatigue, dizziness, movement disorders, tremors, blurred vision, decreased libido, dry mouth, and weight gain. And then tyramine, that's the amino acid I was trying to remember. Tyramine, if you eat or drink something that has a lot of tyramine in it, you're going to become very ill. This amino acid will build up into a very significant level, and it could kill you, actually. So if you're taking MAOIs, you have to be real careful and always check the contents of whatever you're eating or drinking to make sure that it doesn't contain tyramine. So here's a list of some of the MAOs that are on the market. Isocarboxazid is Marplan. Phenelzine is Nardil. Selegiline is Eldepril. And tranylcyclopromine is Parnate, right? And this right here tells you the selectivity. So there's 2 general types of MAO molecules, MAO types. There's MAO type A and MAO type B. So Marplan, for example, works with both MAO type A and B. They all do, except for Eldepril, which is more sensitive to MAO type B. So the research continued from the 60s, right? And by the late 70s, like 78, 79, researchers started to note that there was 2 broad categories of effects related to antidepressants. There was the effect of stimulating behavior, kind of making you more jumpy. And there was the effect of elevating your mood. Okay. So the initial thinking was that the stimulation of your behavior was mediated by blocking the norepinephrine reuptake, so that there was more norepinephrine to make you, get you going, right? And that the mood enhancing effect was mediated by serotonin, right? So feeling better and feeling less suicidal, etc., was due to the effect of blocking serotonin uptake and increasing the amount of serotonin. That was the theory starting in the late 70s, right? And with that, researchers began the process of creating more selective drugs for serotonin reuptake, to inhibit serotonin reuptake, because they wanted to focus more on the mood elevating factors and focus a little bit less on the behavior stimulating factors. And as a result, they came up with a whole bunch of different compounds that we call today the serotonin selective reuptake inhibitors, SSRIs. So we have probably the most famous one is fluoxetine hydrochloride, fluoxetine, which was sold by Eli Lilly as Prozac. Another oldie but a goodie is sertraline, sold as Zoloft. Paroxetine, Paxil. Fluvoxamine is Luvox. Escitalopram is Lexapro. And citalopram is a relatively new drug. This came out in the last 10 years or so, I guess. Celexa. Whoops. And you see here the half-life of these drugs. You'll notice that the longest half-life belongs to Prozac. And there's a little star here which says active metabolite. What that means is that when Prozac is metabolized by your liver, it breaks up into new molecules, broken up into two new molecules. Well, one of those new molecules is still a pretty decent SSRI. So basically, the Prozac gets broken up, and even the metabolites of Prozac are still have an effect on serotonin reuptake. So those molecules have to be metabolized as well. So that's why the half-life of fluoxetine is 24 to 48 hours. Right? It's a long time. Citalopram similarly has a long half-life of about 48 hours, or 45 hours, I should say. Then you have the short ones like Paxil. It's 24 hours. Paxil is very often given for anxiety disorders. And the short half-life means that it won't stay on board too long, so it'll be there long enough to help you deal with your anxiety attack. And then it'll, fade away. Unlike Prozac, which will stay in your body a lot longer. So how do they work? Well, these molecules were engineered by medicinal chemists to block the reuptake of serotonin, and by a competitive binding with the serotonin transporter protein. So normally the serotonin transporter protein binds to serotonin, and it grabs it, and it lifts it, and it brings it back up into the neuron that it came from. But what these compounds do, what these SSRIs do, is they compete for those reuptake sites, and they sit there. And once they are clogging up an uptake site, the uptake receptor cannot do its job, so serotonin remains in the synaptic gap a lot longer. So this blocks the reuptake of serotonin, and the serotonin is left in the synapse for longer durations, allowing it to achieve a longer, stronger effect, and increasing the activity of serotonin circuits in your brain. So that's basically how it works. The SSRI side effects are not as pronounced as the ones associated with the tricyclic antidepressants, but there are side effects. Right, probably one of the biggest ones in males is sexual dysfunction. So erectile dysfunction, basically, decreased libido, GI problems, gastrointestinal problems, decreased appetite, insomnia, and agitation. Right. If you have too much serotonin activity, so if you overdose on serotonin, one of the things that might happen to you is this serotonin syndrome, which basically is a very toxic reaction that is caused by excess of serotonin. right? And it can completely disrupt your breathing and your heart rate, and it can be fatal in some cases. One of the big discoveries over the years of serotonin, particularly Prozac, but of SSRIs, was that they really probably shouldn't be given to children and adolescents. Because the reason was that children and adolescents that were placed on therapy with SSRIs tended to show a very significant increase in suicide and suicidal ideation, which is pretty incredible, pretty amazing. But it took a lot of, unfortunately, it took a lot of lost lives for people to realize that before they started to realize what was going on. So we've seen this picture before, right? Again, we have the presynaptic cell. This is, remember, the action potential is coming down here, causes the serotonin vesicle to move forward and pop, releasing the serotonin. The serotonin then travels across the gaps to these receptors, these little cups that are sitting here in the postsynaptic membrane. Once the receptor is bound to the neurotransmitter that causes a signal, which is represented by these little upside down Wi-Fi icons here, and the process continues, right? There is a reuptake transporter here that is a protein that takes the serotonin back up into the presynaptic cell to be recycled. And that's right there is what is blocked by the SSRI, the reuptake inhibitor. It blocks reuptake so the serotonin stays out there a lot longer, right? This right here is an autoreceptor. Basically, this is a receptor that detects the presence of serotonin. So if there's too much serotonin in the synapse, this autoreceptor will be stimulated like crazy, so that'll send a signal to the cell saying, hey, whoa, slow down. Quit making serotonin. Stop producing serotonin. So over time, what you're going to see is, if taking Prozac over time, you're going to see some changes in how much serotonin is produced, and you might have to increase your dosage. One of the theories of depression is that maybe depressed people have an irregular amount of autoreceptors. So maybe, you've got too many of these guys that are sending a signal to shut down your serotonin. So even if you just have a little bit of serotonin out here in the gap, maybe, just maybe, you've got too many autoreceptors and they're all getting stimulated and telling this cell to quit producing serotonin, which reduces serotonin and ultimately causes the symptoms of depression. So one of the things that the SSRIs do is they downregulate these autoreceptors. SSRIs make these autoreceptors go away. And since they make them go away, that means there's going to be less signals to stop the production of serotonin, so you'll have more serotonin produced. And here is a map showing, they didn't really, it wasn't until the late 80s that there was this link established between children and SSRIs, and it was a big deal. It was a big deal. It was in the news, and I mean, this was a huge deal. That kids were getting more suicidal when they took an antidepressant. So after in the late 80s, they decided to basically stop prescribing these drugs to adolescents and to children. And what you see here on the y-axis is the suicide rate per capita or per 100,000 people. And so the higher number means lots of more suicides than the lower number. And then you have here the years from 1988 to the year 2004. And you'll notice a massive decrease in the number of in the suicide rate. This is suicide. This isn't ideation. This is actual suicide, right? You'll notice a huge decrease going from 1988, 4.4 per 100,000 all the way down to less than 3 per 100,000. So there's a big decrease in suicide after SSRIs were no longer prescribed to adolescents and to children. Later on, as we got better at creating uptake inhibitors, another class of uptake inhibitor that was created were the uptake inhibitors that block the uptake of both serotonin and norepinephrine. And these are the SNRIs, the serotonin norepinephrine reuptake inhibitors. Which is kind of funny because if I'm depressed and I take amphetamines, I'm not going to get undepressed. I might get worse even, but I'm going to be agitated and jumpy, right? But I'm still going to be depressed. I'm still going to have the mood disorder. But if I block the reuptake of norepinephrine, so instead of amphetamine directly stimulates norepinephrine receptors. That's what it does, right? You take amphetamine and your norepinephrine receptors are stimulated. The amphetamine binds to the norepinephrine receptor and stimulates it. That will not cure my depression. However, if I take another drug that instead of blocking the norepinephrine receptor itself in the postsynaptic cell, instead it blocks the uptake of norepinephrine, that seems to help with the symptoms of depression. So these new drugs, SNRIs, are being developed to block the uptake of both serotonin and norepinephrine. So how do they work? They differ quite a bit from the different SNRIs have different effects on serotonin and norepinephrine. And some even kind of have an effect on dopamine activity. And that may have something to do with the fact that stimulating the noradrenaline or the norepinephrine might have a cascading effect of then increasing the amount of dopamine, right? So the SNRI itself is actually increasing norepinephrine activity, and that may have a side effect of increasing dopamine activity, right? So that's a possibility. These are some of the SNRIs that are out there now, right? Venlafaxine, that's the one I take. Trade name is Effexor. It blocks norepinephrine and serotonin. Doesn't do much for dopamine. Nifazodone is Cerazone, which, like fluoxetine, it's got a decent half-life, mainly because it produces a metabolite that is still active, like Prozac did. And nifazodone works on norepinephrine and serotonin, and also is an antagonist to a certain type of the serotonin receptors, type 2 and type 3. which is kind of interesting, right? There's different types of serotonin receptors, and nifazodone seems to block all serotonin, and it has an antagonistic effect on these two guys, 5-HT2 or serotonin 2 and serotonin 3. Remeron or mirtazapine has a bunch of weird effects, right? It blocks the alpha-2 norepinephrine receptor, which is a specific norepinephrine receptor. And it also blocks the autoreceptors for the serotonin 1 autoreceptor. And it also blocks activity of the serotonin 2 and 3 receptors. And then Cymbalta, which is one of the newcomers, duloxetine, it blocks uptake of norepinephrine, serotonin, and dopamine. Just blocks them all together, right? And you'll notice, again, this very, very distinct difference in half-life. I can tell you from personal experience that venlafaxine has a very short half-life. If I miss a dosage, I get severe headaches. But then on the other hand, you have mirtazapine, which has a very long half-life because it does a lot of different things, right? So those are the SNRIs. Then we have these weird and atypical antidepressants, these drugs that were sort of accidentally discovered to have antidepressant effects in some people. These are called atypical because they don't directly work on serotonin or norepinephrine like the other drugs do. They're not like serotonin or norepinephrine uptake inhibitors. They do something else. The most famous atypical antidepressant is bupropion. It is a dopamine reuptake inhibitor. So what Prozac does for serotonin, bupropion does for dopamine. Okay, and believe it or not, in some people, this acts as a pretty decent antidepressant. Bupropion is quite often given in conjunction with another antidepressant. So if you're taking Paxil and it's not quite working for you, and you've tried the dosage and tried other things, sometimes some docs will try, okay, let's try some bupropion on you to see what happens if I give you the Paxil, which blocks serotonin, of course, and norepinephrine, and let me give you the bupropion, which will slightly, let's start with slightly blocking the dopamine transporter and see what happens, right? For those of you that haven't been through a diagnosis of major depression and treatment for major depression, it involves a lot of trial and error for some people, for a lot of people, I guess. I was lucky. I started taking Prozac in 1996, I guess. I don't even remember anymore. But and it worked right away. It worked straight away. And so I was really lucky. But sometimes there's people out there that have to try all kinds of different combinations before they find something that works for them, that doesn't make them, that gets rid of their symptoms, their depressive symptoms, and doesn't have too many bad side effects. So here's some of the atypicals that are out on the market right now. Wellbutrin is probably the most famous one. Bupropion. Then there's Ascendin. You can see what they do. There are different mechanisms of action here. Deserel, which is trazodone. All these different types of drugs that are there. Blocking uptake. Look at the half-life of maproptyline, right, which is luteomil. 51 hours. Blocking norepinephrine reuptake for 51 hours. That's a long half-life. That is a really long half-life. So how effective are these drugs, right? These drugs have made a lot of companies a lot of money. There have been a lot of people prescribed these antidepressants. Do they work, right? Well, the answers are kind of mixed, right? Most of the research out there says that roughly 40% of patients with major depression don't respond initially to treatment. And these are the folks that have to start, experimenting with different types of drugs. And as you can see, you guys have just seen like all the major drugs that are used, and there's quite a bit of them, right? The other bad, well, I don't want to say bad thing. The other, complication. Well, that's the word that's here, complication. The other complication with antidepressants is that a lot of people get well without them. a lot of placebo-treated patients get well as well as the patients getting the drug. So that's an issue. However, if you look longer term, these long-term studies that look at people with recurrent depression over a long period of time, they suggest that over 70% of major depressive disorder patients medically treated improve significantly. Okay, which is a pretty decent number, right? I mean, if you can even save just one person from, committing suicide, I think it's worth it. If you've ever wanted to take an herb that has antidepressant effects, you might want to look at St. John's wort, Hypericum perforatum. It's been studied for a long time and used for quite a while in the treatment of depression. It turns out that St. John's wort does have a effect on increasing serotonin and norepinephrine activity, which is kind of cool. And believe it or not, it appears to do so by inhibiting reuptake of monoamine neurotransmitters, just like the classic SSRIs do. So it seems to do that pretty well. Here's a picture of the plant, the St. John's wort. It's like a little flower. It's like a little flower. And you can see here these effects in this chart here. On the y-axis, we have the HAM-D score, which is like a little test you can take to see how depressed you are. So the lower you score, the less depressed you are. The higher you score, the more depressed you are. So you want to, and it'll, I have thought about hurting myself. I feel hopeless. You answer these questions, and for every one you answer in the affirmative, you get a point. And then they add up all the points. And if you have a lot of points, then that means you're very depressed. And if you don't have that many points, it means you're less depressed. So in this chart here, you will see that along the y-axis, we have study weeks. So how long you were in the study, right? From 0 weeks to 8 weeks. So it's like a little time series here. And you have 3 groups: the Hypericum, which is the St. John's wort, the placebo, and the sertraline, which is one of the atypical antidepressants. So if you look at the placebo group, which is the little circles, let's look at them first. You see, over time, I'm getting a little bit better. And then if you look at the Hypericum group, you will notice they're also getting better. They're a little bit more depressed than the placebos. And then you'll see that the sertraline group is, over time, they're also decreasing better, but they're actually lower. They're feeling much better than the other two, right? And the take-home message here is that the Hypericum does have like a very slight effect, but it's not huge, right? But it does have a slight effect on your depressive symptoms, right? Oops. Oh, let me make that big again. Sorry. There we go. Okay. So that was talking about major depressive disorders. Now let's shift gears and talk about bipolar disorders. These are distinguished from major depressive disorders by the fact that not only do you have depressive episodes, but you also have what are referred to as manic episodes. And during a manic episode, the patients experience a huge elevation, an elevation in mood and euphoria and lots of energy and enthusiasm, which sounds pretty awesome, but maybe not, because what happens with a lot of people is that they get so manic that they do stupid things like overspending money, leaving their spouses, making really bad decisions, all kinds of things like that. These manic episodes can get so bad that they can even lead to a type of psychosis where the individuals are starting to believe these things that aren't true and hallucinating. So it's a rather serious disorder. So the criteria that are used to diagnose a manic episode: a distinct period of abnormally elevated, expansive, or irritable mood lasting at least 1 week. Okay, so if somebody's just in a really good mood for a morning, that doesn't count, right? You have to have this sort of like extreme euphoria for at least 1 week. During the mood, and then you have to have a mood disturbance. And during this mood disturbance, you have to have 3 of the following symptoms: inflated self-esteem or grandiosity. This is during the manic phase, right? During the manic phase, increased self-esteem, like, oh, I'm super, I could do anything. Decreased need for sleep, excessive talking, racing thoughts or ideas, distractibility, increase in goal-directed social, work-related, or sexual activity, excessive involvement in pleasurable activities that have potentially painful consequences. So making a lot of bad decisions, basically. It's possible that, so you have this roller coaster, right? Full-blown manic and then depressive symptoms. And these symptoms can alternate every few days, or sometimes they're present together. You'll be manic one moment and depressed the next moment. A lot of these patients have depressive feelings of hopelessness, even suicide, during a manic episode. And it's not as bad as with major depression, right? They may have some suicidal ideation, some hopelessness, but it's not as bad. But you can have like this mixing. It's not a perfect, depressed 1 week, manic the next week. You can have these mixing of these symptoms between the manic and the depressive. So one of the, obviously one of the first observations of this disorder was the fact that it's cyclical, right? There's like this cyclical nature to it. The depression and the mania just kind of come and go, right? So that leads people to think, well, it's got to be some sort of like real-time disturbances in how your brain is functioning. There's got to be some kind of like shifting pattern of something going on in your brain that is responsible for these cycles, right? Giving mood stabilizing drugs like drugs used for depression has been completely a big goose egg. Not much has been learned. As a matter of fact, it can actually be dangerous to give a person with severe bipolar disorder a drug like, for example, Prozac, any one of the drugs used to treat major depression. As a matter of fact, when you're first getting diagnosed for major depression, one of the things they do is to make sure you don't have bipolar disorder. So if you present yourself to a psychiatrist with major depression he or she can see that you're clearly depressed, right? They'll diagnose you, but then they'll ask you, like, do you ever have like times when you have racing thoughts and you've made some like weird decisions and you can't stop talking and you have trouble sleeping? They're asking, do you have manic phases? And they're asking that because if they diagnose you with major depression, but in fact you're manic, you have manic depressive disorder, bipolar disorder, what'll happen is that the medication that you're taking, like the Prozac or the Paxil, whatever you're taking, is going to increase the manicness. It's going to make you even more manic, which can be very dangerous. The old joke, mental illness is, I got it from my family, right? It's genetic. And bipolar disorder follows a very distinct genetic pattern, right? There are some genes that are related, that are thought to be related to bipolar disorder, but this type of research is very young, very early on, right? This is one of those types of things where there's probably multiple genes involved multiple times in some people and not in others, right? There have been some studies that have found strong correlations between the expression of the BDNF gene and bipolar disorder. So maybe alterations in BDNF production is what's causing this bizarre fluctuation from manic to depressive. So that's a possibility. Folks that have had bipolar disorder for a long time have a pathology in their brain volume. They seem to have really large ventricles. I don't know if you recall, when we were looking at the brain, there were these fluid-filled cavities down in the brain. Those are called ventricles, and they're filled with cerebrospinal fluid. In persons that have suffered from bipolar disorder for a long period of time, their ventricles are enlarged. So if their ventricles are enlarged, that means they have less brain volume. And also, we've seen decreased levels of BDNF in patients with very severe manic episodes. So it appears that the worse that you are in terms of your manic episodes, the less BDNF you seem to have. Okay, so maybe this decrease in BDNF has something to do with the symptomology that we see in bipolar disorder. So here's some data on the brain volume. And we have ventricular volume, which basically means if you're in the middle somewhere, that's good. You don't want to be too low or too high. So the middle is good. So the value of 1 is a good value to be. 1 basically means 100%. This is a healthy control. The healthy controls their ventricular volume. The middle bar represents the ventricular volume of somebody that had 1 manic depressive episode. And you'll notice it's slightly lower than the healthy, which is, that's cool, right? But look at the brain volume of somebody who has had multiple manic depressive bipolar episodes. Their brain volume is almost, not quite, but almost twice that of a healthy brain. So they've lost a lot of neurons. They've lost a lot of neurons in their brains. Maybe related to that decrease in BDNF. That's one of the big research areas right now. So the treatment of bipolar disorders is kind of, again, it was an accident, sort of, where, somebody sort of stumbled upon it, and that somebody was John Cade. John Cade believed that the mania was caused by too many metabolites. like nitrogen-based metabolites, right? So basically, your liver and your kidneys weren't doing their job. Right? They weren't doing their job. There was some excessive amount of metabolic metabolite floating around in your blood that was probably causing the manic episodes. So he had one of his patients. He got the urine from one of his patients and injected guinea pigs with it. And it turns out he would inject guinea pigs with a bipolar urine and urine from non-bipolar people, quote unquote normal controls. And it turns out that more guinea pigs died from the bipolar urine than from the normal urine. So he thought, well, maybe that means that the toxic substance is uric acid. Okay, maybe they're producing too much uric acid. So he figured, okay, well, that's an acid. That's a uric acid. So maybe if I were to give some sort of salt, I can counteract the effects of the uric acid. So he injected his test animals with lithium chloride, lithium carbonate, which is a very simple compound. It's just a carbon and oxygen bound to a lithium ion. And sure enough, it worked. The animals that were injected with lithium carbonate survived more than the ones that didn't. And here's the weird part. You know, nobody knows how this works. Lithium carbonate is a standard treatment for bipolar disorder. It works for a lot of people, but it's a very simple molecule. Like I said, it's just lithium bound to a carbonate group. It's absorbed right straight away into the bloodstream, but it takes a while for it to cross the blood-brain barrier. It's not as lipophilic as you'd like, so it takes a while for it to cross into the blood-brain barrier. It's very hydrophilic, meaning it loves water, but it's not very lipophilic. So it takes a while for it to cross the blood-brain barrier, but it does do so. On the long term, giving patients lithium carbonate is associated with weight gain, thyroid disease, skin rash, kidney dysfunction, and a compromised immune system, which is interesting. Obviously, you would expect the kidney function because you're excreting all of that lithium salt, so, lithium compound, so obviously it's going to put your kidneys into overdrive. And the thyroid and skin rash and immune function would also be expected. It's the simplest compound. It's cheap. It's used to treat many millions, hundreds of thousands of people with bipolar disorder, but nobody knows how it works. We haven't been able to figure out why this stuff is effective in treating bipolar disorder. We just don't know. And part of the problem is that it goes everywhere. I mean, it's a very simple compound that just goes everywhere, and it has multiple effects in many parts of the body. So it's really hard to say what lithium carbonate does to help treat folks with bipolar disorder. Another drug that's used to treat bipolar disorder is valproic acid, and like Depakote. I don't know if you've ever heard of Depakote before. Depakote is it's an anticonvulsant prescribed to persons that are suffering from epilepsy. Depakote is probably the most common one. Also, valproate is out there as well. But valproic acid came about in 1994. I remember very well when it came out. It was a big deal back then. And it was used to treat seizure disorders and migraine headaches. It's also used to treat, can be used to treat bipolar disorders and reduce the severity of the manic episodes. It does have an effect on BDNF synthesis, so it does increase the amount of this BDNF protein that you create, that you can create, and it does alter the activity of several monoamine neurotransmitters. So it does have an effect. It does have different effects that are related to depression, anxiety, and mania and whatnot. But we're still not quite sure precisely what it's doing and how it is helping to treat even convulsions convulsions. Gabapentin, which was sold as Neurontin, was approved in 1993 for the treatment of seizures. It became a very popular alternative to valproic acid and lithium for treating the mania associated with bipolar disorders. The thing about this drug, gabapentin, you'll see the word GABA in the name, GABA. If you recall, GABA is a neurotransmitter that's found in our brains that is primarily used to inhibit neuronal activity. So whenever your GABA receptors get stimulated, they inhibit neural activity. And gabapentin was constructed by medicinal chemists to structurally mimic the neurotransmitter GABA. So, which makes sense, right? If you're having a manic episode, then activating lots of your GABA receptors, which decreases neural activity, should help decrease the severity of the manic episode. And that's basically what it does. So here, the drugs that are used to treat mood disorders, bipolar disorder are listed here. Mood stabilizers and anticonvulsants. So there's lithium carbonate. We know that it kind of works on serotonin and dopamine. We know that much. And valproic acid is a glutamate antagonist, so it blocks glutamate. Remember that neurotransmitter that's everywhere that stimulates activity, it blocks that. And it works as an agonist against dopamine. And gabapentin blocks glutamate and also blocks or stimulates GABA. Pregabalin stimulates GABA. It's a GABA agonist. Lamictal, that's a fairly famous mood stabilizer, is a glutamate antagonist. Try Topamax, another famous drug, glutamate antagonist and a GABA agonist. All these drugs, right? All these drugs. Abilify, that's a fairly new one. That was to treat schizophrenia, and it's a dopamine D2 agonist and a serotonin agonist, which is interesting. So there's your mood stabilizers. There's a whole bunch of them used for treating epilepsy as well as mood disorders like bipolar disorder. And that ends the lecture on Chapter 3. If you have any questions, please shoot me an email, and take care.